everyone. My name is Diallo, and I'm the head of the Solution Science team at Form. My team is responsible of leading scientific and technical pre-sales discussions with our customers, and basically that will include discovery calls to understand their pain points, their challenges. We'll be also doing platform capability demonstration and proof of concept. I'm really excited to be here today to talk about gene and cell therapy manufacturing with my colleague Tseng from PacBio.
Hi, everyone. My name is Liz. I'm an Associate Director of Product Marketing at PacBio. I'm happy to be here.
Thank you, Liz. Great. So the goal of this webinar is to explore what an end-to-end solution for gene and cell therapy can look like. We will start with a quick overview of gene and cell therapy. Then I will hand it over to Dr. Tseng to talk about the PacBio solutions around laboratory preparation, sequencing, and data analysis. I will then introduce the PharmBio platform and focus on our solutions for AAV vector optimization, validation, and data analysis. We will end the webinar with some thoughts on how we can expand our current solution and a Q&A session. Let's talk about the overview. So gene and cell therapy can be defined as the introduction, removal of changed genetic material of patients to treat a specific disease. Those therapies, they can take different forms. So we have what we call gene replacement, where we replace a non-working gene with a healthy one. We have a gene silencing when we inactivate a mutated gene that becomes toxic to the cell. We have what we call a gene addition when we introduce a new gene to target a specific aspect of the disease. And we have a gene editing when we permanently manipulate the gene in the patient's genome. So those therapies, they can be either in vivo or ex vivo. Ex vivo gene therapies involve removing cells from your body. Those usually will be autologous therapies, but now we are seeing more and more what we call allogeneic that are using compatible donor cells. So those cells will be modified genetically and will be placed back into your body. With in vivo gene therapy, new genes are directly inserted into your body by using what we call vectors. For in vivo gene therapy, selecting the right vector or the right delivery mechanism is very important. You can define a vector as the vehicle that we will use to deliver the therapeutic genetic material into your body. And typically, those vectors will be derived from viruses because through evolution, those viruses, they become very, very efficient at infecting the human body and getting into human cells. But we also have what we call non-viral vectors, such as the lipid nanoparticles, the liposome, or also the naked DNA. Vector may have different characteristics in terms of expression, genomic integration, transduction, packaging capacity, and immunogenicity. As an example, if you take the lipid nanoparticles or the nanoparticle in this example, they will have a low immunogenicity, and that's very good because that's what we want in terms of immune system. But on the other hand, they will have a short-term expression. So when you will inject those therapies, they will not last a very long time into your body. Another consideration is the packaging capacity, and the capacity can be greater than 10 kb for those nanoparticles compared to the other ones that we have where the capacity is way lower. Currently, the lipid nanoparticles are the most clinically advanced non-viral gene delivery system. And the fun fact is that you probably have received vaccine using those vectors. So the COVID vaccine that J&J and AstraZeneca manufactured used the adenoviruses vector as a delivery. And on the other hand, Moderna and Pfizer, they use what we call liposome, and those are non-viral vectors. So AAV have been around for a very long time. So it was discovered more than 50 years ago by three scientists named Atchison, Hogan, and Ho. And it took around 20 years to have the first AAV vector for gene therapy in 1985, and another 10 years to have the first in vivo RAAV application and the first human RAAV application. Unfortunately, in the late 90s, in 1999, AAV therapeutics were then slowed down by adverse effects, and then they had the first death in a clinical trial. In the early 2000s, a lot of discovery were made around the AAV capsid, serotype, and tropism, meaning the protein shell surrounding the AAV vector, and the tropism basically is the type of cell that those different vectors will be able to infect. And usually this will help reduce the immunogenicity and also target the cells of interest or the tissues of interest in this case. The first EMA, European Medicine Agency approval, was in 2012 for a drug called Lidera. And unfortunately, that drug was removed from the market for a commercial failure 5 years after because it's really expensive to produce those different drugs and the company wasn't making money. In the US, the FDA or the Food and Drug Administration approved a first therapy in 2017 for a drug called Luxturna to treat patients with inherited retinal disease causing blindness. Since then, multiple therapies have been approved. One of them is Zolgensma to treat spinal muscular atrophy. To date, there are approximately 8 viral vector therapies that have been approved to the US using 3 main viral vectors, the AAV that we'll be focusing on and talking about today, but also the lentiviruses and the herpes virus. There are 25 viral vector therapy in late stage development, and currently there are around 120 in phase 2 clinical trials as in February 2022. So their numbers will keep growing over the next few years, and what we are seeing as a trend will be for the FDA to approve more and more drugs, so the pace will be more and more fast over the next few years. Thank you. So in terms of drug development, this is a process that is very time-consuming, costly, and it contains preclinical, clinical, and commercialization steps. So at a high level, I know those steps are way more complicated, but just at the high level to give you an overview, during the preclinical phase, the research is usually done in animal studies, and we aim to have a better understanding on the toxicity, the activity, the expression level, and this is also where we try to understand some of the early formulations. If the preclinical phase is successful, we continue with the clinical trials involving patient enrollment, human studies, with the goal to make sure that the therapy is safe, the therapy is effective, and we also try to have a better understanding of some of the side effects by using those therapies. In principle, if all those processes are straightforward, a drug development can take a few years, 5 years, 7 years, and so. But in practice, unfortunately, the drug development process will take more than a decade. There are a lot of tightly regulated procedures that we need to go through in order to make sure that the drug is safe and to make sure that the drug is effective. So between the preclinical and the clinical phase or before the commercialization, there will be a lot of interaction with the regulatory agencies in the US and in Europe to make sure that they will review everything and then they will approve those different drugs based on all those different criteria. The solution that we are building here at Form aims to reduce the risk linked to those different therapies, and for our customers, the cost and the time to put those different drugs to market. And currently, we have been focusing on AAV drug development, and I will be talking about that in the second section of my presentation. Now, I will hand it over to Dr. Tseng to talk about the PacBio solutions.
Thank you, Alpha. So I'm going to quickly talk about how PacBio's HiFi sequencing, which is a third-generation long-read sequencing technology, can be beneficial for doing AAV vector design and analysis. So long-read sequencing using PacBio HiFi can be beneficial for different phases of AAV development. Our customers have used, for example, in the initial phases of discovering novel AAV vectors, where they design specific primers to target the constant regions of the AAV vector and sequence clinical tissues to discover novel AAV vectors. In the second phase of the AAV research and development, our customers have been using hi-fi sequencing to look at whether they can identify different AAV designs would result in undesirable fragmentation or truncation of the produced vector. Also, if you look at the little bullet with number 3 on there, long-read sequencing can be applied to other kinds of sequencing as well, including mRNA sequencing. In this case, our customers have also used PacBio to sequence the produced transcripts to see if the design gene actually expresses the desired transcript isoform. And finally, hi-fi reads can be used to validate the production level of different truncation, impurity, and host integration events. Our customers have also used PacBio sequencing to examine the effects of different production systems. This is what our users have been using PacBio hi-fi sequencing for. Starting with a custom designed vector, PacBio provides a AAV library preparation protocol that supports multiplexing of up to 24 AAV samples per SMRT cell. So SMRT cell is our unit of sequencing. Our instruments, Sequel 2 and 2E systems, have a special on-instrument AAV mode that is designed to process the complicated AAV structures. AAV can be in both self-complementary or single-strand form. It is important to properly process them because the sequencing data has to present what the actual molecule structures are. For example, in the next slide, I'll show you that even if you purchase a commercial vector that was intended to produce single-strand AAV, it is possible to see that some of the package genomes are actually self-complementary. Finally, once the sequencing data is produced, we have a bioinformatics analysis code via GitHub right now. But as Alpha will show you later, we're also working to incorporate this analysis with FormBio. So this is a currently released public AAV data set. We've released both a commercially available self-complementary and single-strand AAV. Our sequencing data and subsequent analysis can separate these sequencing data into different types, such as coming from the vector genome, RepCap, helper, backbone, host genome, and chimeric events between any of these quote-unquote genomes. We can characterize whether the produced genome is single-strand or self-complementary. We can also look at whether they're producing full vectors, partial, or backbone. We can also look at the ITR configuration and determine whether the genomes are in flip-flop, flip-flip, flop-flip, or flop-flop configurations. And just to show you an example of what the data would look like, in this table, we're showing with 2 commercial AAV that the characterization, for example, of the self-complementary one, 95% of the sequencing data represent full self-complementary AAV vectors, exactly what you would want to see. For the single-strand AAV, we see that 35% of them are full single-strand AAV vector genomes. However, 29% of them actually produce a self-complementary structure.
Thank you, Liz. Now let's talk about the FormBio solution. So I will talk about the company, a quick introduction. I will introduce the platform, and then I will end with the current solution that we have and some thoughts on the future solutions that we are thinking about building. So at Form, we recognize that organizations are currently facing unprecedented change and opportunity. And we believe in order to be successful, you need to have subject matter expertise in a lot of different fields, such as cloud computing, biologics, AI and ML, gene editing, gene therapy, and so on. And hiring internally all those experts and putting in place an efficient collaboration process is a very hard problem to solve. And what ends up happening is in those companies, they will be asking a biologist to become a bioinformatician, a data scientist, and they will be asking an engineer to play a role of a DevOps. And a lot of the therapeutic companies that we have been working with, their expertise rely a lot on the gene therapy, and most of the time they will need some assistance, some help with all the other aspects of the analysis. The consulting firm McKinsey and Company published earlier this year an interesting study on how to scale viral vector therapies. One of their main takeaways was to consider how digital tools can further unlock operational challenges in production. For example, yield can be optimized with advanced analytic-based models. At Form, we have been building solutions in order to model in silico and optimize the construct before manufacturing. So this really resonated with us. And there is also the link of that study. I think it's really interesting to go through that to understand some of the challenges and some of the thought process that they have about the tools, the solutions that we should be focusing on in order to solve this problem. And how are we doing that at Form? So at Form, we are building a scalable platform, and the next slide will give you an overview of what that platform is. But on top of the platform, we are collaborating with our customers, gene and cell therapy companies, synthetic biology companies, to build solutions for those specific needs that they have. So it's not only a platform, it's also all the advanced applications that we will be bringing on top of the platform to build those end-to-end solutions for our customers. The main goal of our platform is to enable biologists, computational biologists, to run sophisticated data analysis at scale. You can interact with the platform by using our user interface for the biologists, but we also have API capabilities and command line interface capabilities as well. Here are the key components of the solutions that we have built so far. The first component is the data management. This includes data transfer functionalities, data versioning, data provenance, and basically this is where you will be able to understand where is your data, how your data is structured, and have a full provenance on exactly how that data was generated, who touched the data, and so on. The next key component is the workflow execution engine. So when you have all those different data sets available on the platform, we have curated, optimized workflows for cost and turnaround time to run multi-omics data analysis. So those workflows have been verified with our bioinformatics team. We have been benchmarking them using publicly available data sets, and we'll have a lot of white papers publishing how those different results can correlate with other best practices available into the market. So after running those different workflows, we'll have the result management component. This is just an easy way for our users to package those different data, those different results, and make them ready, for example, for publication. So when you think about open science, when you think about reproducibility, when you think about open source, when you publish, being able to be sharing those different data sets, the tools that you use for the other scientists to reproduce and validate and improve those research is very fundamental. And this is also where you will be collaborating internally or externally with other teams. The team collaboration is mainly around providing the right user permission, either at your organization level, meaning everybody involved in your organization, or project level, meaning the specific project that you are working on. And then they will be having the right access to the subset of the data that you would like to share with them. We have been focusing a lot on data visualization. How can we bring in the right visualization tool for the biologists to understand those different files generated, to find insights, and to focus on the science? So when we think about data visualization, we are thinking about bringing integrated genome viewer. We are also thinking about dynamic visualization using ArcShiny application, Jupyter Notebook, to explore the data and then slice and dice it to understand the results. And we are in the process of prototyping, ingesting out of those results into queryable databases to be able to build on top of the platform really advanced visualization tools using all those different data modalities that we'll be able to access. That being said, our main focus is around advanced applications. So the platform will be the vehicle to deliver all those different applications, but we want to focus and we want our main differentiator will be around advanced applications that we are building. When you think about an advanced application, we are trying to solve challenges in product development processes that can be overcome with computational tools and standardization. So as an example, we were spin out from Colossal, and they have been focusing on the extension. So bringing all those the extension pipeline around ancient DNA analysis, around multi-omics analysis and visualization, around CRISPR was a key component. And today we'll be talking about cell and gene therapy, but we have been also focusing on synthetic biology as a whole to understand the bottlenecks, the challenges, and bring our expertise and collaborating with our customers to solve those as well. Our platform currently have the following solutions for AAV candidate optimization, validation, and data analysis. So the first solution is the AAV PacBio QC. This workflow can be used to assess the quality of the AAV sequences using PacBio sequencing. And the second one is the AAV gene therapy candidate optimization. This is a workflow that will be using our pre-trained models. We have different models to optimize the AAV construct in order to reduce manufacturing truncation. The AAV PacBio workflow aims to characterize, as Liz was saying previously, the AAV PacBio sequencing dataset and produce metrics and visualization reports. As an example here, you can see on the left side some information about the distribution of the reads. So it's really important after the sequencing to understand if my reads were mapping basically to the backbone, to the transgene, if I have a full construct versus a partial construct, if I have chimeric reads, understanding where those chimeric reads are coming from in order to optimize the manufacturing process. And we are in a process to optimize and update the pipeline that we have by including the latest improvements that Liz added to the PacBio workflow. So you will be able to characterize even further those ITR region with the flip-flop and so on. So please stay tuned on those updates, and you will be able to run that tool out of the box directly on the PharmBio platform. Manufacturability is critical, but unfortunately, during the preclinical phase or early clinical trial phase, the main focus is around efficacy and non-manufacturability. And this leads most of the time to design flows that are not captured, and the results will be in very low yield and dose when you start your manufacturing process. So there is a real challenge in order to optimize gene therapy design before the manufacturing and bring in those concerns as early as possible in the drug development process. Especially what we have been seeing is a lot of those therapies have been moving from ultra-rare disease to rare disease, and now we will start seeing more common disease for those different therapies. So being able to increase the dosage for each of those manufacturing on will be critical, because that will enable our customer to reduce the cost of goods, the cost of production. As an example, when you are running a manufacturing run in those bioreactors, you are talking about tens of millions of dollars in order to run a single run. And sometimes the amount of doses is really low. So if you can double or triple the amount of dose, you'll be able to reduce the cost. And on the other hand, you'll be able to serve more patients that are really in need of those therapies. So on the PharmBio platform, we built an optimized model to optimize the design in silico before starting your manufacturing process. So our current model includes herpin analysis. This will allow us to determine alternative but chemically equivalent nucleotide substitutions to make constructs more resilient to truncations. We have CpG island detections, where we enable the usage of SOs that do not trigger an immune response from non-methylated CpG island, because a lot of the problems are linked to the immune response. So if we can lower the immune response, that's a big win. And we have also the codon optimization functionality. So what we are trying to do is basically to fine-tune constructs in multiple different parameters simultaneously to improve the manufacturing while maximizing the safety and the efficacy. Because if you are able to produce more full viral particles, you will need to ingest a lower dose, and ingesting lower dose will reduce all the risk to trigger the immune system at the end of the day. So we have a convolutional neural network model, and our machine learning model is based on understanding the positioning of those different features, understanding the sequencing, and we also try to represent those sequences in 2D and 3D. So we try to understand the energy characteristic between them in order to build the model. And this model has been used by our customers to predict multiple constructs that are more effective at being fully manufactured. So for one of the therapies that we are able to analyze, our model predicted 70% reduction in truncation in the CDS region, meaning the coding region. And when we received that construct was already optimized internally by our customer. So this was a very promising type of prediction. So if you take a look at the image on the left side, so the blue dot represents the original sequence when we received it, and the orange dot will represent the optimized one. And then on the y-axis, you have the truncation proportions. So by running our model, we were able to predict a new construct while basically we will have the exact same protein produced to have way lower amount of truncation. And this was really a promising result. So prediction alone, they don't mean much. So we are in the process of validating those predictions. So we are collaborating with a lab in UT Southwestern to scientifically validate those predictions. So in our design, our validation methodology consists of growing the original sequence that we received. We'll also add a control sequence in that run. We are going to use one of the commonly open source tools to optimize constructs, just to compare it to what we are doing. And then for our model, we'll use it in two ways. So one way will be to minimize the truncations. This is what we want. But the other way that we wanted to test it as well to make sure that the model is understanding the causes behind having truncation and contamination, we want also to be able to maximize them. So those sequences will be tested in living cell for accuracy. Those constructs will also be sequenced following the PacBio protocol that Liz presented earlier and analyzed with the QC pipeline that we already have on the platform. On top of that, we'll be running potency assays. The potency assay is critical because this will allow us to confirm that the protein that we will generate will be functional, and then those proteins will be able to be expressed properly. So when we'll have all those different results, we'll be publishing a white paper detailing all the process and all the outcome that we will get by scientifically validating those predictions and compare the predicted model to what will happen in reality. So we talked about the current solutions that we have, and I believe there is a lot of promise in terms of optimizing the construct before the manufacturing, and also just providing an out-of-the-box solution to understand your AAV PacBio sequences, because a lot of the gene and cell therapy companies that we are working with, their subject matter expertise is on gene and cell therapy and less on bioinformatics and running those different workflows at scale. So being able to give an out-of-the-box solution was key. But we want to think beyond that. What are the other aspects that we should focus on in order to provide an end-to-end solution? So we see a lot of value in AAV capsid selection and optimization. So the capsid serotype and tropism can determine the severity of the immune response and also the type of cell that will be able to infect. And as you know, most of us may have been already infected with some AAV serotype. So once you are infected with AAV, what will happen is your immune system will build some immunity mechanism. So the next time you have the same infection, you will trigger an immune response. So usually before providing AAV therapy to a patient, they will test him to make sure that he wasn't already infected with that specific serotype, Otherwise, we won't be able to take advantage of that therapy. And then the other aspect of it is once you receive a dose, it's less likely that you'll be able to get a second dose. So you have only one shot. So being able to have capsid that can be used for more use cases that will reduce the immune response is part of what we think an end-to-end solution should look like. The second aspect of it is for AAV optimization. When you have your vector, we want to have a better understanding of everything happening from ITR to ITR. So all the regulatory elements, how can we improve the promoter regions? How can we improve the enhancers? Thinking about the UTR and all the other aspects of that vector and see how we can have a better construct as a whole. We have been currently focusing a lot on the CDS regions because the work that we have been doing was with some therapies already in clinical trials where changing the construct a lot will require going back to all those regulatory agencies and then running a lot of comparative type of analytics. But if you are able to collaborate with customers early enough in the drug development process, we'll be able to take a look at the promoters and all those different elements, recommend an optimal construct before moving to the clinical trials. And we are actively looking for collaborators to run and improve those different tools that we are building. The other aspect of this is to expand the solutions that we have toward ex vivo therapies and also toward the other platforms, such as the adenoviruses, the lentiviruses, and so on, rather than AAV. AAV has been one of the most used. That's why we have been focusing on that, but there are also needs in order to take a look into the other vectors available. We have been talking, and then we are in early conversation with CDMO, contract development and manufacturing organization, in order to help them optimize the construct that they are receiving, but their main focus is around manufacturing. And most of the time, there is no standardization around those processes. So how can we optimize the construct and then work with them on the downstream to be able to optimize and standardize their manufacturing processes? So understand what are the optimal parameters in order to have, at the end of the day, a higher yield. We believe that even if we optimize the construct, if the manufacturing process is not optimized, we won't be able to take advantage of all those different improvements. Last but not least is around all the interaction with the regulatory agencies. So this is a clear bottleneck from our discussion with our customers. It's time consuming. So we are thinking about how can we help create a report, standardize the report, and integrate those reports with the right regulatory agencies. That will help save time and also reduce human mistakes at the end of the day. And by using our platform, if you are able to do your capsid optimization, vector optimization, manufacturing, everything will be tracked, versioned, provenance will be included. It will be relatively straightforward to generate as needed those different reports for all the questions that the FDA or the EMA will be able to ask you, and then be able to submit those reports directly to those organizations. So we think that there is way more that we can do on the field, and we are really looking for collaborators to tackle those challenges together.
Okay, so first question, looks like this is for Alpha. How can I access the PacBio platform?
So currently we have, when you go on our page, on the PacBio platform page, there is a waiting list. So you can submit and we can add you to that waiting list, and somebody will be able to follow up with you over the next few days to give you access to the platform.
Thanks so much. Okay, next. So this looks like another question for Alpha. Do you support any common workflow languages?
The short answer is yes, and that's a very important question. So we decided to build all those workflows that we have so far on top of Nextflow. because NF Core and the community has been contributing a lot into building standard workflow to be utilized. But we recognize as well that there are other standards such as CWL, WDL, WDL. So we have into our roadmap on how can we add those other workflow languages and then create integrators to be able to run those workflows directly into our platform. So on our roadmap, the first priority is to be able to run out of the box the workflows that will be available in NF Core or any Nextflow workflow that you will have in your GitHub or Bitbucket repo.
Thanks for that. Okay, so this is a PacBio question for you, Liz. So what other applications besides AAV sequencing can benefit from PacBio long-read sequencing, and how can users access PacBio sequencing?
Yeah, that's a really good question. So as I said, PacBio is, long-read sequencing can work for anything like gDNA or cDNA. Our customers have been using PacBio, for example, sequencing the plasmid. They've used it to sequencing the mRNA to see what transcripts are being expressed, and of course the AAV vector itself. And in terms of access, PacBio is not a sequencing provider. We actually sell our sequencers to sequencing providers.
That's great, thanks. Okay, another question for you, Alpha. What cloud providers do you currently support?
Yeah, that's an interesting question around cloud providers. So currently we are on top of GCP on Google Cloud, and we started to focus with Google because they have a very strong partnership program with startups and small companies, and we are going to move towards enabling, first of all, on GCP, our customers to bring in their own ID. So if they already have an account, they will be able to use their own storage, they will be able to use their own compute resources. But we want to extend it next year to the other major cloud providers such as AWS and Microsoft Azure. We have a lot of current customers that have been asking for that, so we have that into our roadmap when we'll be able to bring in your own ID. And what I want to add as well is we have been seeing customers also not asking for a cloud provider, but being able to use their internal resources. So a lot of them, they have their own HPC. And then we are also investigating and prototyping how we can enable our customers to use any type of compute and storage. It can be a cloud or their local infrastructure to take advantage of them. And we'll have more detail on our roadmap over the next year. And if this is a use case that you would like us to focus on, feel free to reach out. We are really looking for early adopters, people to share that same vision to build really a solution that will solve those different needs. So if this is something interesting to you, feel free to reach out, and then we'll be more than happy to connect and start brainstorming on what the best solution will look like.